So for the remaining of this course, really, spanning to the end, we're going to start to cover something very particular, and that is a set of libraries from the Bioconductor consortium, and they allow us to interact with these types of data, and they have functions and methods to allow us to process and analyze this data as we may desire. Okay, so we're in our Intro to Bioconductor page now, and we have our integrity checks, our setups, many more packages to install. So please get going on and trying to install these then. I think we actually need base core QC. You can download the material here. We're going to start with this first presentation, and this is just a introduction, a love letter to Bioconductor, so we can all be as excited as I am about this. Because Bioconductor really is a consortium which has changed the ways we can interact with data, particularly genomics data. So this is the introduction to Bioconductor. Bioconductor, BioC, often shortened to, is an open source. It's important it's open source. We know exactly all the code in there. Community-driven software project. So it isn't necessarily, there are people employed by Bioconductor, but really it's people like you and me. We will write these software, we maintain them inside the Bioconductor consortium's framework. Okay, so they provide then sets of tools and databases, we, the community, for the analysis of biological data. Okay, so we are biologists, so we do biological data. And really, although I say biological data, a lot of this is focused on first expression arrays, then all arrays, and now sequencing data. So it started in 2001, and it started with the R from R, him and Rossi Harker. So Robert Gentleman was more of a biologist, and he ended up starting Bioconductor. And it gained popularity really through two microarray packages, Lima, which we still see nowadays, from Waikato, University of Waikato. This is a really great standard for microarray analysis. Murray was also super popular at the beginning, but never moved into high throughput sequencing, which Lima adapted to. So gained popularity, core reviewed. This is really important. There are people making sure, and this is main job, really maintaining the integrity of the packages in here. It's all versioned, so we're never going to have to worry, what version did we use? Why can't I repeat this result? And now, it used to be different. We're going to have two major releases of Bioconductor a year. I think R used to have two as well. Now R only has one, but they used to be in sync. But now you're going to have two Bioconductor releases, two sets of modern packages being added every year compared to just R getting just one. What's really good and why we're here is that it has fantastic support for high throughput sequencing, really fantastic. And I started learning, I was doing microarrays during my PhD. So I really witnessed all this high throughput sequencing environment and functions and libraries all build up. And it's been really incredible. So the broad goals of Bioconductor project are to provide widespread access to a broad range of powerful statistical and graphical methods for the analysis of genomics data. I'm not sure genomics data is their only limit now. They have proteomics data. They have other types of data in there. But for sure, they want to provide this range of statistical graphical methods to visualize your data too. They put a lot of effort into things like including biological metadata into the analysis of genomics data. So they have packages, libraries which contain annotation, information from literature, which you can use in your analysis. And then to build really a set of standards, but a common software platform that enables rapid development, deployment of software, which is interoperable. So they try and encourage you to have software which talks to other pieces of software in Bioconductor, so we can all work with each other, and it's very scalable, right? So as we'll see as we move forward through this course, we can use this on big data too. Really important things about Bioconductor, which I think separates it from CRAN or the standard R repositories, is they really concentrated on high quality documentation as well as reproducible research. last thing they really set out, and this is a big goal of Bioconductor, is they want to train researchers too. So they want to make their workshops available, their courses, and they put this all online on their websites so that other people can learn how to use these tools and then perhaps go and write their own tools and then train other users. So this is what Bioconductor looks like as a website, kind of stretched. I think I have it open. So this is what Bioconductor actually looks like right today. We have your about, your news, install, so guides on how to install stuff, lots of guides on how to learn stuff, really using how you can use this in everyday solutions, but also other people using community events. And then for developers, if you want to write your own packages, you can come here. This has got lots of useful information, how you set up your creating a package, how you test it, etc. A lot of us will be here and install and learn, right? Also other things, events, Bioconductor 2021 coming up, worth everyone going to. Okay, so that's the Bioconductor website. So it can be broadly split into 4 groups, 3 main groups, with one group kind of expanding now. And that is software. So you have Bioconductor packages which contain tools which allow us to actually analyze the data or visualize it. We have annotation. So these are tools for the integration of this biological metadata. You know, this symbol means this symbol in a different place. So, you know, I can map my entree symbol to something I can read, a gene name, or an ensemble symbol, so I can swap it with someone else who works in a different database. The functions associated to a gene, all these are stored inside these annotation data. And then what's really nice is they actually have experiment data as well. And this isn't full experiment data, but they have data sets which are big enough to make you use, to really encounter real world problems, running out of memory, takes time to actually process. They can put this inside what they call experiment data packages. So you can actually download the data from a particular publication, perhaps. Last thing they have, and I think this is a really lovely addition, but going slow in Bioconductor, is their workflows. So they have workflows which really talk you through how you would do an analysis using a combination of software and annotation, and maybe by using some experimental data. So this is really what puts it all together, and this is really the learning aspect, right? Workflows. So you'll often find a workflow for the most common data types, or the most fashionable at the time, but they quickly fall out of date. And this is one problem, I think, with the workflows on Bioconductor. Don't think that's right. I don't think we're in that's the latest build of Bioconductor. I might be wrong. But for sure, it has over now 2,000 software packages, and all these packages are peer-reviewed. So we have the Bioconductor community will review them, but also one of these central people in Bioconductor will have a look at this particular software and see if it really passed the standards. It is tested and evaluated automatically. Any changes which happen in the Bioconductor ecosystem are propagated to the libraries, the other things which depend on them, and all this is tested and evaluated when any changes make. It just means Bioconductor is super robust. When you download it, you can be pretty sure it's going to work, as long as you're using the release version. If you're a developer, maybe not. What's really important is actively maintained and updated. So, you know, you're expected, part of signing up to Bioconductor, part of putting a package there, is you're expected to ask questions, and you're expected really to update your package and keep it passing these tests as the years go on. It isn't frozen at that particular point. And that's where these great things like versioning are very important. If we're going to maintain and update it, it's great that Bioconductor has version releases, which allow us to go and say, okay, I was using that version, I will stick with that because that was doing what I expected. The new version uses a slightly new method. Maybe I'll use that in the future. Before being accepted to Bioconductor, and I kind of just said this, all new packages have to pass this review, and this tests the manual and all examples. So everything they say in their manual, every example they say they have as an illustration of how to use it, must work. Okay. At the same time, people do go through and pick on the way you code in order to try and make your code a little bit better. So this is what it looks like. There is an open review site. We can go there, and we're going to go spy on. So, you know, Matt has a package. Herper, he's going to show you this. Doug, who's going to come in next week, he has Profile Plier, and we have other few packages knocking around, but one of the ones I wanted to show today was rfastp. So this we submitted last year and it got accepted, but you can really see here, you know, you have to accept that I'm going to do this work. They come in, they have a go at your description file, and you can read through, these are all open. You can read through all the efforts which people go through, all the automatic testing, the comments, before eventually, you know, someone says, okay, this now meets our standards. And there are 63 open right now, but there are 1,880 which have been reviewed. A lot of these may be rejected or multiple times to the same one. But I think this is great, and I think it's great it's open. So anyone can hop in and review. Anyone can actually see how a package is being developed. So it's useful if you actually want to develop a package as well. So this ensures they all build on the latest version of R, right? So an old package, which you download an old version, may not work on the latest version of R, but every time a package is released, they know it is working on the latest version of R. The examples in the reference manual can be evaluated. They actually have to work. And your vignette, which is your use case, talking people through how to use the package, not just examples, but really talking them through the lower levels. All this has to run too. Okay, so I've got base call QC, which is an older package, but we can look at what this looks like. For any bioconductor package has to have these standards. And I really think this is what raises it above R and CRAN and some of these other places. So every software package is listed here that we can start from the beginning. So if I go install, I can see we have all the older versions if I needed to, previous releases. We have software, annotation, experiment data, and workflow packages here. If I want to, I'm just going to click on my software packages. You can see them all later. You can see that there just aren't that many workflow packages. There is a nice one on single cell though. And in fact, Aaron Lund's written a great book on this. I was going to go look for rfastp. So, you know, you could Google this. Or actually, maybe I should just Control F search it. Here it is. It's written by Wei, maintained by me. And we can see it passes all platforms. Not very popular yet. We've only just released it. Builds okay. Importantly, we can see here some instructions on how to install this. We have our manual here, PDF, and we have our HTML description of the vignette here. Okay. This is a nice package. Every package in Bioconductor gets this page. It's automatically built and it's automatically tested anytime anything which could break it is changed. Okay, so I'm not quite sure. Yep, let's talk through this and then we can go back to the actual, see it in life in the example there. So every package has this little install command, so you can just copy and paste this from here. And if you want to, then it's just going to go install first a manager from CRAN. This is BioC Manager written by Levi. And once that's installed, we can make sure we have the right version. This is wrong, 3.10 should be whatever our latest version is. And then we install our Bioconductor package just using, rather than install.packages, we use BioC Manager, : install, and we list the package we want. Again, I don't need to actually remember that. I could just copy and paste it from here, and that will work every time. You're meant to just copy and paste it from there. All the dependencies, every other package I use, this package hasn't invented everything from scratch. We use other people's software. All that gets imported, all that gets managed by, not just a clever name, BioC Manager. Okay, so you don't need to worry about what version you're on. Once you've installed, as long as you don't mess with your BioC Manager, you can just carry on installing your software using things like this. If you ever need to change your version, and I strongly recommend you don't do this, you can do BioC Manager install, and then you set your version. Okay, I say you strongly say you don't do this. Don't do this if you're unsure. Okay, you want to maintain your versions for the entirety of your project, really. Okay, so then all packages have this reference manual. So I can actually show you the FastP reference manual. Where was it? Okay, so we all have a reference manual. This is the install. If I click here, and this is automatically built from the actual code, we have all our functions which we can use inside this package listed here in the index. I can click and jump to them, right? Tells us what the actual function name is, and it tells us the input, right? What parameters I need to provide, what the defaults are, if there are any, and then it describes what actually these inputs are, right? Get the description, this argument means this input file should be a vector of input file names. And then finally, we get some examples which will run without needing to download anything else, right? They use data built into the package. And you can be sure that this is a good example of how to actually run the package and get a result. Okay. Yeah, so these are nice detailed examples. We know that everything is going to work in there. I think something which is also super important is the vignette. And here we have one which was written by Wei in entirety. And it's here that you describe more stepwise what people should perhaps be doing with your package. You know, what is your package generally? How do you install it? What would we like you to do with the package, right? So this is more of a story about how to use it. The reference manual is more the instructions, the blueprints of how each function works. Cool. Great. So that's a little bit about all the packages, or how you would start to review and get some of these packages and how these packages are set up. They all look the same. If I want to install this one, I copy this. If I want to know how it works, I might choose one of these vignettes here. And if I want to know how the actual examples and the functions work under the hood, I would go use this reference manual. I think it's this uniformity across Bioconductor, this forcing people to really buy into their way of doing things, has made Bioconductor such a success. Okay. Why we really like Bioconductor on top of all this is that it allows us, as I said at the beginning, to cover a wide range of biological data types. We're mostly focusing on high throughput sequencing. Arrays probably done, maybe SNP arrays still. But we're going to be focusing on the high throughput sequencing type of formats, and in particular these common data formats we've already described. FASTA and FASTQ, we looked at these earlier on. SAM and BAM, the aligned versions of FASTQ, once I've aligned it to the genome. Can we handle those in R? We can. Different variations of the bed files, the bed6 that has names, strand, and score, or encodes narrow peak and broad peak, just the extended versions of bed6. And also our gene models, we want ways of handling them in R2. Great. So now I can just start to show you this, really, just give you examples. So genomic sequences, these are our FASTA, our identifier in a long string of sequence. These are handled very neatly in the biostrings package. And it's actually what we're going to see today. Genomic intervals stored as bed files are handled using the R track layer package. This gets them in and out of R. And it's the genomic ranges package, which has the functions to allow us to start to operate on these genomic intervals. For WIGs or bigwig files, remember these long stretches of steps of scores, or the bigwigs, which is the compressed version. We can use our track layer to, again, import them, export them, and genomic ranges, again, like with bed, because they're very bed-like, allows us to kind of interact with them, manipulate them, analyze them. GFF features, you can start to see a trend here, genomic ranges, genomic features. GFF files, these are our gene models. These are handled using the genomic features package, which just extends genomic ranges to now take into account some of this additional metadata information. Remember, these exons belong to this gene. FASTQs, like we showed earlier on and what Connie was producing for us, These are handled using the short read package because they are short reads. Okay, so both import, export, manipulation is handled in short read. SAM and BAM files, they're handled using this genomic alignments package, which is a real boon. And actually, I haven't mentioned it, but import export's handled by something called RSAMTools, which is the R equivalent of SAMTools, a very popular library. It's genomic alignments, which really allow us to interact with these. All this information you see in the file, it's genomic alignments, which allow us to extract this and manipulate this. As well as software packages, which allow us to import, export data, manipulate this data inside R. We know Bioconductor maintains lots of annotation packages. These include things like microarray annotation. This probe matches to this part of the gene. Gene to ID mappings, gene functional annotation. So when we want to do things like gene set enrichment analysis, Go enrichment analysis, we come along, we need to know this gene is in this particular function. This gene does this action. And really important for any basic processing, we have genome sequence information. So all this FASTA we described, which we can get from iGenomes, there are packages which actually maintain this internally. On top of that, we also get the equivalent of a GFF, our gene models, within this gene or transcript. We can also get those from inside Bioconductor too. So first thing to look at is gene annotation. We have a set of libraries which allow us to extract the annotation of genes to symbols, genes to entrees IDs, ensemble IDs, functional annotation. And these are all contained in what are org.db packages. So actually, I think if you open Bioconductor, you can find a subset of assay packages called org.db. Okay. Now, the format for your particular species of interest will be org, species of interest, ID type, and then DB, database, right? So for example, a human, we have org, we have HS, Homo sapiens, EG, this is entrees gene, and most of, if not the vast majority of these databases are using entrees gene, and then .db. Okay, we can see they have a ton of these, 20 species where they've done all this mapping, ID to entrees ID for you, to gene ontology for you. And we don't need to have a different version of this for every genome build, because this is just the mapping of gene names, right? Genes to functions. So this isn't changing in combination with the genome builds, GRC37, GRC38. Okay. But talking about genome sequences and genome builds, if we want to get the full genome sequence from a file, we can use these BS genome packages. And we have for every or most of the major species, we have BS genome, the species of interest, the source. So where did they actually download this from to get this sequence? And then this major version. So, GRC38, HG19, the major revision of the genome. So for instance, for HG19, we have BS genome, species is H. sapiens, the source, and most of the sources are UCSC. We do have some, if you look through, which are ensemble, and then we have the major version here, HG19. So this is a slightly older build of the human genome. HG38 is the new one. On top of sequences, we also can get gene models, and these are within our TXDB packages, our transcript database packages. And here we have a very similar format again. For our genome, it's going to be species, source, major version, and finally we have this idea of table. So this is Homo sapiens gene build from UCSC. So it's going to be TXDB, H sapiens, it was HG19, it was from UCSC, and then actually this known gene. If you go to UCSC, and maybe we'll have time at the end, you will find a reference to a table called known gene. Okay, and this little description here tells us exactly where it's come from. These files themselves, these packages, internally also contain this description. So we can really review where all the information came from. Oh, wow. So we've got time for an exercise, but I think I'm going to skip the exercise because it's fairly straightforward, and we'll pick this up at the end. for when we have for when we start to go through the sequences. Okay, so I'm just going to ask you to install a package in this point anyway.